And what's more interesting is that these tumors that occur in people with the breast cancer gene mutation, but who get pancreatic cancer, they look the same on a CT scan. But if you look at the genetic abnormalities, they're quite a bit different because of that breast cancer mutation. And just in the past couple of years, scientists have started wondering maybe they can target the mutation or the abnormality in the breast cancer protein in these pancreas cancers. And there's drugs called PARP inhibitors, and there's some very old drugs called cisplatin that was thought to work in the test tube. And I can tell you the name of this patient because she's publicized it all in magazines, and it's Libby Znaimer, who's Moses Znaimer's sister. She's about 52, and she has hereditary breast cancer. And the story was actually in the Star, so it's perfectly ethical for me to tell you the story. You can see it. You can read it yourself anyways. So she actually came to us 2 years ago with pancreas cancer. And on the CT scan, it was not operable. It was invading a big artery called the SMA, or the supramesenteric artery, and you can't take that artery out successfully. You can't live without that artery. But because she had the BRCA2 mutation, her medical oncologist decided to give her what's called tailored chemotherapy. The drugs that we normally give may not work, but he gave her a different kind of chemotherapy. And we actually saw that tumor shrink away from the artery, and it became operable. And in 20 years of doing this, I've never seen that. We see a lot of people who come to us with inoperable pancreas cancer, and we give them chemotherapy, we give them the hope, but the reality is it never happens, or I've never seen it happen. But this is the first time it happened, and we operated on her. It's 2 years ago in July, and she's still free of disease. And I think it's because of this. You can see the benefit of the molecular biology of understanding this disease, and that's a pancreas cancer patient that normally would have not had a happy ending. I don't know if it's a permanent happy ending, but it's certainly, for the time being, a happy ending. So what you'll see in the next, what you'll probably hear, as well as you read about this every day, is what's called personalized tailored therapy. And what we're trying to do is tailor the treatment to the patient's cancer, not assuming that all the patients are the same, and that's where the significant advances are appearing. So I think I'll stop there with that particular story and pass on the mic.
Thank you, Steve. That was a great introduction. I'm Sheila Singh, and I'm a pediatric neurosurgeon at McMaster Children's Hospital. I'm telling you this because I want to tell you the very first sort of observation that made me realize that the conventional therapies that we use right now on patients, surgery and chemo and radiation, work well for a certain subset of patients, but really distinctly fail on another subset of patients. And so when I did my training here in Toronto, I met two little boys when I was doing my pediatric neurosurgery rotation at full kids, 5 years old, and they both had a type of childhood brain tumor called medulloblastoma. And this brain tumor arises in the back of the brain, can grow quite quickly, and always requires surgery to remove it first, and then we follow up with chemotherapy and radiation. That's our gold standard therapy. And when you combine these three therapies, you actually get an increasingly better and better survival because of really good surgical techniques and microsurgical techniques that enable us to take the tumor out better now, because of really refined ways of delivering very focused radiation therapy so that it really just targets the tumor and only millimeters away and you don't damage normal brain, and advances in chemotherapy in delivering more selective and better drugs. And this has raised the 5-year survival for this tumor to almost 90% now. But the 10% of children for whom it fails, and the ones who don't respond, have a very bad outcome still. Not to mention that the side effects of all the therapies Dr. Gallinger and myself are mentioning are huge. And especially when you're radiating the brain and giving these drugs, there's obvious neurocognitive impacts on the life of the child. So basically, I saw these two little boys. They were treated with the exact same therapeutic regimens, and they had the exact same tumor. Same pathology. When you looked at it under the microscope, it looked exactly the same. And yet, one little boy flourished, and he successfully came through therapy, and he's probably going to university, but the other little boy died. And the question that brought to my mind is, for everything that I'm learning in my surgical training, what's the difference between these two boys? Because they had the same tumor, they had the same gold standard therapy. And so what's the biological difference between the two? And that's where I decided to pursue scientific training, because I realized that the answer probably lies in the realm of molecular biology or in the realm of genetics. So basically then, what I want to tell you from that is that led to some research that we did to try to define cancer stem cell populations in brain tumors. So I'm now just going to try to explain to you what the cancer stem cell hypothesis is in about 5 minutes. And I'll try to define it for you. I'm going to try to tell you why it's so exciting to clinicians and why it's such a clinically attractive hypothesis. But then I'm also going to tell you about some of the caveats of it. Because like any new finding, any new thing that comes up, and I think I can't tell you how many times I've heard this described as like a sexy topic. Cancer stem cells are sexy. Everyone thinks it's sort of exciting and attractive, but the point is that sometimes there's a lot of hype around things that are considered current or exciting. And so I'm going to tell you a little bit about some of the caveats of the application of the cancer stem cell hypothesis to all cancers. So the cancer stem cell hypothesis very simply implies that there's a population of cells within a tumor that have stem cell properties. And stem cells are the primitive cells that give rise to all of the different organs in our body. And you may have heard of embryonic stem cells that give rise to the entire person. We're not talking about those stem cells. We're talking about somatic or tissue-specific stem cells. So for example, liver stem cells would be able to give rise to every cell that makes up the complex organ of the liver. And neural stem cells give rise to all of the different lineages that make up the brain. And so with this, stem cells are actually very powerful cells because they have the capacity to renew themselves and to generate all of the cells that make up an entire organ. So the cancer stem cell hypothesis simply states that there are some cells within the tumor that behave like stem cells. It doesn't imply that means that cancer originates from a normal stem cell. What that means is that cancer can originate from a normal stem cell, or it can originate from a progenitor cell, or a fully mature cell that has decided to go back and reacquire stem-like properties. So one of the caveats about the cancer stem cell hypothesis is it doesn't immediately mean that, oh, that means cancer comes from stem cells. No, it means that scientists have made an observation that there's a group of cells that somehow have stem cell properties that could arise from a normal stem cell within a tissue or organ, or they've reacquired those properties because of perhaps some mutations that they've accumulated. So that brings me to why should stem cells be the target of cancer in that case, if you do believe that cancer does arise from a normal stem cell. So for example, I do believe actually that brain tumors arise from a normal neural stem cell. And why would I think that rather than any other cell within cancer? The reason is because stem cells actually are one of the rare types of cells that are around for the entire lifetime of an organism. A lot of cells have a very rapid turnover rate. Blood cells only live for 120 days, and some cell types turn over even faster. But stem cells are technically there from the moment you're born and in varying numbers until you die, presuming that in your brain you don't burn them all out by doing a lot of drugs or drinking a lot of alcohol. So basically, if I do believe that, then what that means is all the genetic mutations that Dr. Gallinger is studying could accumulate much more readily in a cell that's around for a long time to accumulate those mutations. Secondly, I just told you that stem cells are really powerful, right? And they have all these abilities to generate pretty much an entire tissue or an entire organ. So imagine that you have a stem cell that acquires a series of mutations, and it really is literally the topic of our talk tonight. It's a good cell that goes bad. What happens then is you have an incredibly powerful cell that hijacks the normal machinery in a normal stem cell to then generate a whole bunch of abnormal cells which make up a tumor. So you can really think about a cancer as an aberrant organ. So if a stem cell generates a tissue or an organ through a process of very finely and tightly regulated divisions and over time and space, these processes that we see in development that are so beautifully regulated of how one cell gives rise to a whole tissue and organ. Imagine now that one cell goes bad and it degenerates into a very disorganized bad tissue or a cancer. So those are some of the sort of principles of why the cancer stem cell hypothesis is attractive. because if you look at it, it's really just that processes of normal development are somehow altered to give rise to a cancer. So what substantiates that even further is that a lot of the things that we find wrong in cancer and a lot of the abnormal genes and signaling pathways are actually all very important to normal development. And some of the key targets and signaling pathways we're looking at are actually genes that are active in normal development. So for example, there's a molecule called Sonic Hedgehog, and everyone likes it because it's named after a video game. So hedgehog is actually a molecule that's very important. It's a morphogen, and what it does is it actually helps to lay out the whole pattern of the spinal cord, and all of the motor neurons that make up the spinal cord are dictated by a gradient of this sonic hedgehog. And so it's a very organized process. In a very organized process, hedgehog helps to determine the patterning of your whole spinal cord, your ventral spinal cord. And so similarly, hedgehog is actually elevated in a lot of these medulloblastomas that I've told you about. And hedgehog signaling is actually aberrant. So what I'm saying is a lot of the processes that we see in normal development are actually dysregulated in cancer. Lastly, some of the therapies we're talking about, chemotherapy, radiation therapy in particular. Not surgery. Surgery is kind of good all around. We take out a lot of tumor and the patient feels better and it's great. But then the therapies that we follow up with, chemotherapy and radiation therapy, they generally target very actively proliferating cyclin cells, cells that are rapidly turning over. And so what that means is if you have a very quiet cell that's gone into quiescence and decided not to divide, then the chemotherapy and radiation therapy will not catch those cells. Those cells will be able to hide out somewhere either around the edges of the tumor or where the tumor was before and evade these therapies. And then that same patient is going to come back and present with tumor again that's recurrent. Right? So basically the cancer stem cell hypothesis explains very well why tumors come back after surgeons take them out completely. It's because stem cells themselves are quiescent. The ones at the very top of the hierarchy are very quiescent. And because they can hide out after a tumor's been resected, they're there to regenerate that tumor again. And so it comes back to what Dr. Gallinger said about delivering targeted therapies. For us, it's not only about individualizing the therapy to the patient, it's also about targeting whichever cells within that heterogeneous complex tumors that are made up of all kinds of different cell types. There may actually, out of all of the cell types in the tumor, only be 1 or 2 cell types that we have to worry about killing, and that may be the cancer stem cells.